In this video, we are going to cover ion channels. The learning objectives for this video are to become familiar with how an ion channel may be activated and appreciate how different ion channel signaling pathways have major physiological roles. Ion channels play major roles in a wide variety of physiological functions such as pain, nociception, thermoregulation, osmoregulation, vascular tone, and endothelial function. They also contribute to the normal function of cardiac, respiratory, urinary, skeletal, digestive, and central and peripheral nervous system function. The more discovered about ion channels, the more they are implicated in disease processes including hereditary channelopathies, induced diseases such as skeletal bone dysplasia, neuromuscular diseases, and motor and sensory neuropathies. As you know from studying cell biology, many processes, from neuron action potentials to pacemaker cell rhythms, require the electrical current created by the flow of charged ions. Ligands combine ion-specific channels and alter open and closed conformations, while some ion channels open after effector proteins are activated, and others are voltage-gated. Cells may have different types of ion channels which allow for tight regulation of intracellular current. Ion channels have very specific pores that allow only certain ions to pass through based on their size and charge. There are several ways to categorize ion channels, including how they are gated. Leaky channels are not gated and therefore open randomly and allow ions through. These are critical for allowing potassium to equilibrate in cardiac cells and create a resting membrane potential. Chemically gated ion channels are controlled by ligands. This includes cation channels controlled by acetylcholine at neuromuscular junctions. Mechanically gated ion channels open their pores in response to stretch and when on a neuron result in an action potential. These channels can help sense many sensations including vibration, pressure, touch, sound, taste, and even vision. Voltage-gated ion channels can be found on presynaptic neurons and open in response to a change in membrane potential. Here is a visual representation of the types of ion channels we just discussed. Cells use several types of ion-gated channels together to create an overall beneficial response. In the next few slides, we will cover a couple examples. Here is an example of a synapse. A synapse is where one nerve axon terminal ends in very close proximity to a second neuron's dendrite, cell body, or axon. The neuron releasing the chemical signal is called the presynaptic neuron, whereas the neuron receiving the chemical signal across the synapse is the postsynaptic neuron. An action potential moves down the axon of the presynaptic neuron, creating an electrical signal that opens up voltage-gated calcium channels, which in turn cause the release of pre-made and stored neurotransmitters into the synaptic cleft. These neurotransmitters might be dopamine, serotonin, acetylcholine, or any number of chemicals. Since the postsynaptic neuron is so close to the presynaptic neuron, a high concentration of ligand is then available to open ligand-gated ion channels on the postsynaptic neuron and continue the signal propagation. For example, an action potential may be triggered on the postsynaptic neuron. Cells have ligand transporters and enzymes to uptake and break down ligands to remove the ligand from the synaptic cleft in order to stop the signal. A very specialized synapse is the neuromuscular junction. This is where a motor neuron ends at a muscle and can cause contraction. Here the same principle of the basic synapse described in the previous slide applies, but instead of a postsynaptic neuron, there are muscle cells. An action potential travels down the motor neuron, opens voltage-gated calcium channels, releases acetylcholine. Acetylcholine chemical-gated cation channels open, resulting in voltage-gated sodium channels to open on the muscle cells, and muscle contraction occurs. Botulism toxin prevents the release of acetylcholine from the motor neuron, resulting in flaccid paralysis. To gain a more rounded understanding of the nervous system and how ion channels can play a role, take a look at crash course videos on YouTube. Here are a couple links that may be beneficial to you. In summary, ion channels form pores that can allow the movement of nonspecific cations or specifically one ion, such as sodium, calcium, or potassium. Ion channels are critical for the formation and maintenance of membrane potentials and electrical signals. Since ion channels are so ubiquitous and control everything from emotion, memory formation, heart rate, and motor function, what issues do you think could arise from a toxin that overstimulates a calcium channel? If an ion channel loses its ability to be selective for certain ions, how could this result in a diseased state? These overarching questions are great to think about as you read and research specific receptors and signaling pathways this week, and learn about the different types of ligands that can bind receptors.